Yes.
Yes, sir.
Share your screen, Prabal.
Okay. Is it visible?
Yes.
Yes.
Okay.
Just go slides for more.
Hmm. So can we start now, or we should wait for some more time?
Yes, 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 sir. You can start.
Okay. And how much time I have?
Sir, up to 4 p.m. 1 hour for 1 hour.
Whatever you will take, but better to finish by 4.
Yes, yes.
Before 4.
Okay. Just 1 minute. Sir, I am in meeting in a presentation. Can I call you back after some time? Okay. Okay, sir. So we just started the fluorescence part. So this is the light source that we discussed already. Now regarding the fluorescence, how it works actually. So we have a filter block. So inside the filter block, we have a particular excitation filter, which is this one, and we have a dichroic mirror, and we have a emission filter. So let's say we have a light source of mercury or LED based light source, and when the light is coming with the excitation filter, we can determine what band pass should go to my sample. And from this dichroic mirror, actually, uh, just one minute, let me change to laser pointer. From the dichroic, this excitation light is reflected to your sample, and then from the specimen, whatever emission is coming, that is finally going to your eyepiece. And before that, we have a emission filter through which we can determine the bandwidth which we want to see through eye or image with the camera. So this is the filter block with which we can determine the excitation wavelength range and the emission collection range. Now if we talk about the filter block, like the filter block that I mentioned in my last slide, it looks something like this one. So you can see this is the excitation filter through which the light is coming, and then this is the dichroic mirror and going to the sample, and whatever emission is coming, it is passing through the dichroic mirror. So dichroic mirror has a particular property. I can hear some noise in the background. Uh, is it repeating or something? Okay, so you can see this is a dichroic mirror, and dichroic mirror has a property. Normally, it actually reflects the shorter wavelength and transmit the longer wavelength. So let's say if we use a DAPI filter, let's say the cutoff is around 450. So what will happen? Whatever light we are sending, let's say around 420 to 440, that is shorter than 450. So this will be reflected, and generally DAPI emission is, let's say, from 460 to 480 that we want to collect. So that will transmit through the dichroic mirror, and finally through the emission filter, we can select with what wavelength range we want to collect. Now, if we talk about in the light path in the microscope, with respect to microscope, so this is fluorescence illumination, and from here light is coming, and this is the filter block. So you can see this is the diptic mirror through which the light is going to your sample, and whatever emission is coming, that is either going to your camera, or there might be a small optical fiber which is going to your longer than 400, that will be transmitted, and BA means barrier filter or the emission filter. So that will allow the 435 to 485 range to be passed through for eye observation or imaging. So that is how from each filter block you can understand what is the specification of the filter for excitation and emission. Now, when you work with any particular fluorophore, you have to first check whether your filter block is matching with the excitation maxima or emission maxima of that fluorophore. If it is not exactly matching with the excitation maxima or emission maxima, at least it should cover most of the wavelength or the
Please, please share your
hello.
screen. Your screen is not visible, sir.
Oh, okay. I don't know why. Sorry. Can you see now? Hello. Hello. Hello.
Yeah,
Can you
it's
see now?
visible. Yes, yes, it's visible.
Okay. So
Carry
now,
on.
so that is how the filtered specification should match with the flow for excitation and emission to get the best output from the from the fluorescence microscopy. So this is the basic part of the fluorescence microscopy. Now, if we want to, let's say, capture a multi-channel image, so how we can do that? Let's say in the microscope, so I showed you this microscope. So let's say if we have some option where I can put multiple filter here. So let me go to again laser pointer. So let's say if we have a particular hardware component where we can put multiple filter, which is called filter turret. Inside there will be generally 6 slots are available. So each slot you can put different filter. So let's say if I have 3 filter in 3 different slots. So first I put DAPI filter to capture the DAPI excitation, to capture the DAPI emission, which generally goes to the nucleus. Because it is binding to the DNA, so it will show you the new position of the nucleus. And then let's say I change either with the hardware component. So microscope might be a fluorescence, sorry, might be a motorized microscope where we can, with the software, we can very easily change the filter position, or we can do it manually. Also, for manual microscope there are different options available. And we can put a green filter, which actually, let's say, as I told you, phalladine alexa fluor 488. So what will happen? We can see the actin fibers, because phalladine only binds to actin fiber, and since the phalladine is tagged with a fluorophore which is called alexa 488, so excitation is around 480 and emission is coming around 500 to 550. So I can see the green emission, and I can see the position of the actin fibers, and then I can use some other filter block to see similarly, let's say, for the mitochondria. So if I do something like that, and I can capture each image with each filter, and then with the software I can merge all the three images, then what I will get, I will get a merged image of all the different fluorophores. And from that a single image, I can understand where is the position of the mitochondria, where is the position of the actin fibers, and what is the position of the DAPI or nucleus. So that is how multi-channel image can give you the information regarding the position of the structure that you are particularly interested, or the position of the organelles that you are interested inside the cell. And also you can see the morphology of the organelle, how it looks, let's say before treatment or after treatment. So this one example where, let's say, this is the light source and we put a optical filter, and by changing the filter position, I can select what is the excitation and emission range, and I can capture three different image. Let's say first one is a DAPI, which actually showing you the position of the nucleus. And then I think that the, I don't know what is these green dots. Let's say this is another protein, and the red one may be actin fibers. So when we merge these three channels together, the fourth one, that is the merge image, or it is called multi-channel imaging. Here we can actually understand how the proteins are distributed inside the cell. So this will give more information, and sometimes you can also evaluate how your protein are like interacting or with each other or not. This will give you some idea, some direction. Okay, so from the multi-channel image you can get this kind of information. Now, this is just one video to show you how advanced now the light source has become for the fluorescence microscopy. So these are small example of prior, which has 16 different wavelength or 16 different LED with different wavelength, which can give you the excitation, different wavelength range for excitation. And you can switch the LED very fast so that you can do very fast imaging. So if I play the video, let me go back to again. Oh, no. So you can see there are 16 LEDs, and each LED excitation is mentioned here. And you can actually use any LED, like four groups are there. You can combine any LED of these four groups, and then you can do the imaging with these four color. Sorry. So here actually you can switch on all the 16 LEDs to have a white light, which you can use for some purpose, or you can individually switch on different LED to have different color. So all these options are possible, and this switching time now with the available new LED illumination is in microsecond. So you can understand how fast you can switch the different color of the LEDs, and how fast you can do the imaging part. Okay, so now hope you understand this part, and then we can go to the next level. Uh, okay, so now with the fluorescence actually, for the imaging part, we have very advanced cameras also. Earlier, actually, the cameras are based on CCD, or EMCCD technology, but most of the recent cameras are now based on CMOS technology. So, what is the difference between CCD and CMOS technology? It is beyond it is difficult to explain here now, but just to give you an idea how, like, when the photon actually comes to the camera chip, it got transmitted into, like, transformed into electron, and then how you are, like, you are taking care of the electron movement from one pixel to another pixel, and how you are doing analog to digital conversion. That actually differs between the EMCCD or CCD based technology and the SCMOS technology. Okay, but just to tell you briefly, the SCMOS technology is now very much advanced. So you are getting with the recent cameras quantum efficiency of around 95%, or there are some option of 95%, and you are getting a resolution of 2K by 2K, which is very difficult with CCD camera. There is I think maximum available up to 1K by 1K, and also the speed is very high, around 70 to 100 frames per second at 2K by 2K. So you are covering all the three parameters that is difficult to reach. One is, like, you are getting better sensitivity, quantum efficiency, better speed, and also better resolution. So that is how the fluorescence microscopy, you can actually capture a very low light imaging also. So that is these are the different parameters that I want to discuss, which are helpful for the, which is actually advantageous position for fluorescence microscopy. Now we will come to a position where, like, we will discuss about why we need a confocal microscopy over a fluorescence microscopy. So this is just to tell you that with the modern day microscope, which is mostly motorized, all the components can be controlled with the just click of the software, and you can actually program the movement of the components. And with the strong software combination and hardware combination, you can do multi-dimensional imaging. Like, you can do a single plane image, you can do a Z-stack, you can do a time lapse image. Like, if you want to do a live cell imaging for 1 hour or more than that, then at, let's say, every 10 minutes you want to capture some images to see how dynamic uh, incident is changing inside the cell. So that you can do. So all these things can be done with the software, and it is you can program the how to run the experiment, and just with the single click it will continue the imaging without any intervention, user intervention. So that is how the modern day microscopy can help you, particularly the wide field fluorescence. Now, what is the advantage of confocal microscopy over wide field fluorescence? Now, as I mentioned, that for the fluorescence microscopy, we use something like mercury illumination or LED illumination. So, if you see the spot of the light size, the spot size of the LED light, it is quite big. And as a result, what happens? Like, if you want to illuminate your sample, since the spot size is quite big, it illuminates a wider area of the sample. Now, let's say I want to image only a part of the cell. Again, I go back to the laser pointer. So let's say this is I want to image only this part of the cell or this part of the sample, not all of the sample. But since the spot size is big, it is actually it is actually illuminating the whole sample, and as a result, it is creating a phenomena which is called photo bleaching. Photo bleaching means sorry. So fluorophore molecule will give you excitation and emission. Now, if you illuminate the fluorophore molecule with like light for longer time or strong amount of light, the fluorophore molecule actually lose its property for fluorescence or capability to give you fluorescence, and it enters a particular state. It is called dark state, where it will lose the fluorescence property. And the more you illuminate your sample with the fluorescence light, more number of fluorophore will go into the dark state. And if you see in eye, you can see gradually the intensity of the sample is going down. The intensity of the fluorescence is going down. That is called photo bleaching. And unfortunately, once the fluorophore goes into the dark state and lose the fluorescence property, it is almost not possible to bring it back to the fluorescence state. So that is the problem of photo bleaching that is associated with fluorescence microscopy. Now, how we can solve? Instead of like illuminating the whole sample, we can illuminate very specifically a part of the sample. That is possible with the confocal microscopy, where we can use a laser because the laser is very like a pointed light source, and this light source actually we can illuminate a part of the sample, and whatever emission is coming from that part we can capture. So in case of fluorescence microscopy, we are using generally LED and Merkel illumination, so we can see the emission with the eye. But since in confocal microscopy we are using laser, we can't see the emission in with eye, because it is hazardous to the eye observation. We use some particular detector for the detecting the signal. So this is the first point of confocal advantage of the confocal microscopy over the fluorescence. So like you can specifically illuminate a particular part of the sample. Now the second part is that so let's say when we do wide field fluorescence, so light is going to the dichronic mirror, it is going to the sample, and whatever emission is coming, that is actually reaching to the detector. Maybe this is a camera detector. Now what happens? This light, this is the focal point where we focus the light. Now, ideally, the condition should be that the emission signal from this focal point only should reach to the detector to get a proper focused image. But that is not the case, because light is traveling along the z axis of the sample, and as a result, the flow force, that is actually above part of the bottom part of the sample, that is also getting excited and give you emission signal, that is also coming and going to the reaching to the detector. And as a result, the in focus and out of focus information both come to the same detector same time and make the image quite blurry, or the signal to noise ratio is not good. You can see, let's say this is my sample where I want to see the small puncta kind of structure. So here, due to the background, it is very difficult. But in case of confocal, we have a pinhole. Probably you have heard about that. So pinhole is a physical aperture which is placed at that point. Now, whenever the light is coming, whatever the out of focus light, that is actually getting discarded because of this pinhole. Only the light coming from the focal point that can reach to the detector. Now, why it is called confocal? Because this pinhole is placed at the conjugate focal plane of this focus point. So that's why it's called confocal. And as a result, since we can discard the out of focus light, you can see the signal is to noise ratio, or the background is much reduced. Signal to noise ratio is improved, and we can see the minute structural details very clearly. So that is another advantage of confocal microscopy over wide field fluorescence microscopy. Just 1 minute. Sorry. Now, if you compare between the two images, left side is the wide field, right side is the confocal. So you can see how the confocal can give you the minute structural details which will be helpful, which can be helpful for your particular experimental question. So that is how confocal is advantageous over wide field fluorescence. And particularly if your sample is quite thick, then this out of focus light problem actually affects the final image more. So this is one example where the Drosophila embryo is stained with cytox green, which particularly stain the nucleus or the DNA molecule. And as a left side, since there is lot of out of focus light is coming due to the wide field fluorescence imaging, you can see only a part of the sample. You can see the nucleus clearly. Other part is actually masked by the out of focus light. But if you go to a confocal imaging, you can see the different nucleus position all over the sample very clearly. So that is why the confocal microscopy is helpful. Now, if we talk about the confocal light path, this is a very simplified light path. So you can see this is the laser light source, and there is a first dichroic mirror through which the light is reflected, and there are two scanning mirrors. So scanning mirrors is like a normal mirror with some particular coating, so that is moving like with a motor, so it is moving like that. So one mirror is projecting the laser along the x-axis, and there is another mirror which is actually projecting along the y-axis. So it scan a particular row pixel by pixel and comes to the second row first pixel and complete the row, and then third row, fourth row, something like that. So probably you have seen in confocal imaging, we generally capture 512 by 500 resolution. So 512 pixel like rows and 512 columns. So and we can scan pixel by pixel with a point scanning confocal microscopy. So this is one scanning mirrors, and this is the second scanning mirrors, and by coordinated movement of these two scanning mirrors, we can scan the entire sample, and this laser is going through the scanning mirrors to your sample, and whatever emission is coming, that is moving along through the scanning mirrors, and finally, since the wavelength range is longer than the dichroic cutoff, it is transmitted, and before the detector we have the pinhole which discard the out of focus light, and finally the in focus information is captured. So this is very simplified light path of a confocal microscope. Now if we see actually in the instrument part, so this is a microscope platform which is called Nikon TI2E, and here on the left side we can have the confocal scan head. So inside the scan head we have many optical components. So here we have to understand here actually inside that scan head we have all the components that I mentioned: the fast dichroic mirror, the scanning mirrors, and also the pinhole. So you can see there are two type of scanning mirrors here, which is called, one is called high resolution scanning, which is a slow scanning mirrors, slow moving scanning mirrors that will give you very precise scanning and very good image quality. And there is another scanning mirrors which is called resonance scanner that actually move very fast. So it is meant or it is designed to capture very fast imaging for fast cellular dynamics, okay, which is difficult to capture with a slow scanning mirrors. So this is the scanning mirror position. This is the laser input port and output port, and and this is the pinhole system, which actually there is a variable pinhole which can actually change the diameter based on the wavelength and the objective you select, and it can collect the information with by after discarding the out of focus light as much as possible. So that is the design of the scan head that we can see on the microscope left board generally. And another very big advantage of confocal microscopy is that we can capture the information along the z layer of the sample. So let's say, so light is focused at a particular focal point. Now if we can somehow move the focal point of the light along the z axis of the sample, what we can do, we can scan a particular plane of the sample, then we can store the data in my computer system. I can go to another layer, I can collect the information. So I can collect layer by layer and I can store the information, and after all the layers are complete, we can combine the information with the software which is called 3D volume view or 3D reconstruction, or sometimes they call volume rendering also. That will give you the idea how the molecules is distributed inside the 3D volume of the sample. So this is one example. Sorry. So here you can see these are the microtubules and chromosomes. So how the chromosomes is actually arranged along the microtubules, we can see in a 3D condition. And also right upper side you can see the red proteins, how it is distributed inside the green protein molecules that we can see. So that is how the 3D view can give you more information regarding the distribution of the proteins or the organelles. Okay, so if we talk about the advance or advantage of confocal microscopy, so you can do multiple colored imaging, and as a result, you can evaluate a colocalization parameter for different proteins. So colocalization means it will give you some idea that let's say A and B proteins are there. So how likely A and B can come together with each other. So this will colocalization parameter can give you some direction or some idea about that, but it is not a confirmed proof that A and B are interacting with each other or not. Okay, so this is not a confirmational test, but this will give you some direction. And since we are using laser light, so this very specific wavelength, so we can do multiple color scanning. And we can do scanning sequentially one after another. So as a result, sometimes what happens if the fluorophore molecules having overlap of the emission spectra, if we do simultaneous imaging, so one signal from one fluorophore can be detected into another channel. But in case of confocal, we can scan separately. I can and we can collect information for one fluorophore at one time without the breakthrough issue. So that is another advantage. And then, as I mentioned, that without physically touching your sample, you can collect the information along inside the along the z direction of your sample, and you can combine them to have a 3D view of the particular sample. So that is how the confocal microscopy can help you for different type of application. So these are the general application available with the modern day confocal. And since we improve the signal to noise ratio by discarding out of focus light, it will improve the resolution, particularly the Z resolution, compared to white field fluorescence. Now there are some advanced application also. So I'm not going into all the details, like FRET or Foster resonance energy transfer. Probably you have already studied it. And then there are two spectroscopy technique, FCCS or FCS, fluorescence correlation spectroscopy, and fluorescence cross correlation spectroscopy, that you can use to see protein-protein interaction. And also there is another technique which is called FRAP, or fluorescence recovery after photobleaching. So these are the advanced application that is possible with the confocal. So here I will very briefly discuss about FRAP with just one slide. So you can see what we do. As I mentioned, that if we illuminate a fluorophore molecule with a high light, it will go to permanently dark state. So it will not give any more fluorescence. So we are using the same methodology here. So what we are doing, we are making a small RY in the software. We first scan one image or take one image and make a small RY. And with the software hardware combination, what we can do, we can illuminate that small RY with very high laser power. So whatever molecule inside that RY, that will get bleached. Man, there is no more fluorescence signal. So you can see this is the RY. So inside the RY, there is no fluorescence, but outside the fluorescence signal is there. Now, if I have a live cell, so what will happen inside the live cell? The protein generally diffuses, or there is some guided transport also that I'm not going into details now. So if the molecules somehow move, so what will happen if we continue the imaging with time? Every time, like out a particular time interval, if we continue the imaging, we can see that the outside fluorophore molecule which having fluorescence property, that will gradually diffuse or move inside this RY, and with time probably we can see this RY which is actually going totally dark, it actually recovers the fluorescence property because of the outward movement of from outward to inward movement of the fluorophore molecules inside that RY. So that property is called fluorescence recovery after photobleaching, and that we can use for studying the different dynamics or diffusion rate of the proteins. And if we change somewhat with different fluorophore is available, which is we can do photoactivation and photoconversion experiment also. Sorry. So these are the some application example. So as I mentioned that we can capture beautiful images with the confocal, particularly multi-channel image. So these are zebrafish example where we have three different fluorophore H42 which generally stay in the nucleus, GFP, Alexa 555. Oh, okay, sorry, there are four colors, and Alexa 647. All the fluorophores are there. And this is one example where published a paper on coronavirus. So they first performed a electron microscopy for the coronavirus, and then validate the data with the confocal imaging. So here you can see this is the electron microscopy image, and they actually did some co-localization analysis with some different viral proteins to see the interaction or whatever finding they get from the electron microscopy. This one example where inside the cell the red color actually showed you the microtubules, and the green one is called EB1 protein that travel along the microtubule. So this is done in a live cell situation. So just one minute. So if I play this one, you can see how the EB1 protein actually traveling along the microtubule. So this kind of dynamics you can do imaging with a confocal, because you can do it without out of focus light. And this is one example of frap. But just notice this part. Now you can see it is getting bleached, and quickly it is also recovering, because it is a cytosolic protein and the mobility is very fast, and it is moving from outside to inside the RY quite faster. So you can see this is the RY where we bleach, and initially the fluorescence property got lost, and after that, with from the outside the protein molecules are moving, and we can see the fluorescence is getting recovered. So that is how the frap can give you, from the frap you can measure the movement property of the molecule. Let's say you make a wild type and a mutated version of the protein, and then you can check how the molecules are, or protein molecules are moving. Okay, so this part actually already I have discussed. And now, as I mentioned, that we with the scanning mirrors, again we can go to, with the scanning mirrors we can scan pixel by pixel along the rows. So that is called raster scanning. And since we are doing raster scanning for a point scanning confocal, that is actually slow scanning, and it takes a lot of time to complete one image. So the disadvantage of having this point scanning confocal, here you can see we have a single variable pinhole. The diameter of the pinhole can vary as per the wavelength and the objective. And since the point scanning confocal is slow and laser is spending per pixel some time, which is called pixel dwell time, as a result the photo bleaching is high. So if we want to image some sensitive sample or life cell imaging, where we want to image without the photo bleaching, or as low as possible for the photo bleaching part, then maybe we can go for some other technique. There is another confocal microscopy technique which is called spinning disk. So this is a point scanning confocal. These are pros and cons. In case of spinning disk, the advantage is that we can still have a pinhole, but here the pinhole is fixed. So confocality is less because we have fixed pinhole, but imaging speed is very high. So this is very good, particularly for sample where you can see lot of phototoxicity. Okay, so here we are using a particular disk where we have array of pinhole, and uh, when it actually spin very fast or rotate very fast, we can very quickly. So this is the pinhole which help to scan along this part of the sample. Then next pinhole will scan the next row. Third pinhole will scan the third row. So something like that. It's rotate very fast, and we can scan very fast for a particular sample. Now, with the modern day spinning disk, the rotation speed can go up to 15,000 RPM. So you can understand, like we can capture even at microsecond exposure time also. And for a spinning disk, we are generally using a camera for high sensitivity. So this pinhole spinning disk confocal, it is first conceptualized by Paul Nipkow in 1884, and later on David Eger and Mojimir Petron actually in 1967, they just make this instrumentation part to make it functional for particular imaging with spinning disk. So this is the light path. Here you can see this is a spinning disk part, pinhole array disk. And sorry, to improve the laser percentage or laser efficiency, there is another disk which is called micro lens disk. So they are connected by a shaft which is rotated very fast in a coordinated manner, and laser is passing through this one through the objective finally to the sample, and whatever emission that is finally from this dichroic mirror is going to the camera for detection. So that is how the spinning disk confocal works. But please remember, this pinhole are fixed diameter pinhole. You can't change the diameter or vary the opening of the pinhole. And also here we are using camera for imaging part. In point scanning confocal, we use PMT or photomultiplier tube for the detector. Here we are using camera. So the advantage of spinning disk is that it is capturing very fast, very gentle to live cell. So that's why sometimes people, particularly who are working with sensitive sample, they prefer spinning disk confocal. Now this is one example where you can see particularly they want to see the calcium spikes. So if you see the calcium imaging inside a cell, it gives you a sudden spikes. Okay, and that is very fast, and it is very difficult to capture this from normal point scanning confocal, which is slow scanning. But it is possible with the spinning disk confocal because it captures very fast. So this is just one example how the spinning disk confocal can help you to capture this fast dynamics. Now I'll come briefly to a particular conception. So when we talk about microscopy technique, so if you want to use, let's say, fluorescence microscope or confocal microscope, you have to understand what kind of detail you want to see for your sample. So there are some basic concept regarding the diffraction interference and how it creates the optical resolution limit. So you can see, probably you already seen this kind of situation in the environment. So this is happened due to the diffraction through the cloud. Now light has a property when it travels along a particular opening, then it creates a wave front. And if we have two different wave opening, like somewhere else, somewhere here where we have two different opening, two wave fronts are created. Now these two wave fronts are merging with each other in a positive and negative interference manner, and as a result there is positive interference and negative interference, which can gives you, if you put a x-ray plate or some film which is photosensitive, you can see this kind of pattern. So bright light, dark region, bright light, dark region, something like that. And if you put a big x-ray film, you can see this kind of concentric circle. So it is similar situation when you put a torch on the wall, you can see some concentric circle similar to that. So you can understand. So this is a small light source, and when the light is traveling along the optics, and you have this kind of opening, so there is a positive and negative interference, which finally gives you this final image of this light source. Although the light source is very small in reality, but when you capture the image, due to this phenomena, it becomes broadened. Okay, so this is called convolution process. And now if you think about two situation where let's say you have two different light source which is very close to each other. Okay, so before that, just think about the situation. So if you capture image in xy plane, it will coming like this concentric circle. And if you somehow manage to capture the image in z direction, so you can see this become like a ellipsoid. So the point illumination, which is like a small sphere, that is no more a small, become a, like you can't capture this small sphere due to the diffraction and interference. Whatever captured by as a final image, there it become elongated like a ellipsoid. So this is the xy view, and this is a yz view. Okay, and now if you think about a situation where you have two light source, so if you project place the if you now put a graph of intensity versus distance from the center, you will get something like this that something like this curve. And now if you consider these two light source or two fluorophore molecules, if they are quite far from apart, like 500 nanometer, then you can separate them easily. But if you bring the these two molecules gradually close to each other, after a particular point, you can see this will no longer be two different peaks. It seems like it is coming from a single entity. So this is the resolution limit beyond which you can resolve the two entity as two different objects. And since the diffraction and interference and is an inherent property of light, so any optical microscope like fluorescence, white field fluorescence, or confocal is bound by this inherent characteristics, and as a result, the confocal microscope resolution limit is around 200 nanometer in XY, 200 to 250 depending on the wavelength and the objective NA in XY, and Z around sorry, so Z is around 700 to 800 nanometer. So if anything closer than that, you can't resolve them separately. So if we that idea first introduced by Lord Rayleigh, first Lord Abbey, and later on modified or fine tuned by Lord Rayleigh. And here, this is the XY resolution, and this is the general formula. So you can see 230 nanometer is the optical resolution limit for a normal optical microscope for a fluorescence or confocal, and this is the Z resolution limit. Now, if we so for if we now want to understand in terms of microscopy scale. So if you want to capture the whole cell or internal organelle, the whole organelle, let's say whole mitochondria or whole ER, something like that, probably we can work with or manage with the normal white field fluorescence or confocal microscope. But if you want to capture more details like internal membrane structure of the mitochondria or internal whether the microtubule is a solid tube or there is an internal hollow, so that kind of details if you want to capture, then it is not possible with the confocal microscope. That you have to for that you have to go for a particular technique which is called super resolution microscope scopy. And for that, I think in 2016, Stephen Hill and some other colleagues also get a Nobel Prize. So nowadays, that super resolution microscopy is becoming too much uh, too much more familiar for application or for experimental purpose. So people, it becomes more and more popular because the like super resolution microscopy earlier it was very difficult because there are to align the optical components to capture a proper super resolved image what was not very user friendly. But with the advancement of the hardware and the software combination, we are going towards more easy capture of the super resolution micro imaging, and that's why it's become more and more popular. So this one example where we can this is a confocal based super resolution. It's called InSpart. So what it does, it has a sorry. So here we use a array of detector, 25 array of detector. So each detector works as a point to unit of pinhole. So generally, when we do confocal imaging, we use one AU pinhole opening. Okay, one AU, one AR unit. That is some value is in micron. That actually pinhole diameter. That much we open to capture the image without to reduce the out of focus light and capture the proper image with the proper intensity. Now, with the InSpark, what we are doing, we are capturing the image, keeping the one AU pinhole so that the brightness of the image is maintained. At the same time, since each small detector uses a zero point 0.2 ARE unit, it improves the resolution. So, we first captured the image, and then we do a Cepheid summation and optical reassignment. So if you search in Google Cepheid summation, there is a very old paper published by Colin Cepheid. That is a very family, like popular or very famous paper. And based on that, there are multiple technique already designed. So so you can search about that. I am not going into that details, but the advantage of having this confocal based super resolution is that you can go up to 100 nanometer in resolution and Z around 300 nanometer, and you can do deeper imaging. Generally, with the super resolution, it is difficult to do deeper imaging, but with the this kind of technology, you can do deeper imaging. And if we talk about the image clarity, so left side you can see this is a nucleus part, and we have this mitochondria captured with normal confocal, conventional confocal. You can see we can see gross structure of the mitochondria, but we can't see internal details. But if we go to InSpot, you can see somewhat details of the internal structure of the mitochondria. Similarly, since you can do deeper imaging, you can do tissue imaging. So this is one example. Right side is a conventional confocal, left side is the InSpot, and you can actually zoom any part of the image, and you can see the structural quality. Right side you can see for this structure it is very difficult to understand whether there is a solid structure or inside there is an opening. But on the left side in Spark image you can see there is a hollow inside. So these are like two different structure come together. So that is how the super resolution microscope can give you a better understanding. And there is also some other super resolution technique available commercially. One is called SIM, or structured illumination microscopy, where we use a particular pattern to generate that super resolution microscopy. And also it's called another technique which is called STORM, or stochastic optical reconstruction microscopy, or PAM. These are based on localization technique. And also there is another technique which is called STADE, or stimulation emission depletion. So these are the three techniques that is commercially available. Just want to show you one image. So left side you can see these are confocal image, conventional epifluorescence, or I think epifluorescence microscopy image. On right side you can see the SIM image, and you can see the clarity of the actin fibers. So that is possible to see. And so the SIM or InSpark, they improve the resolution capability twofold compared to the confocal microscope. Now if you want to go more, then you can go with a stone, which improves tenfold optical resolution. Means you can resolve up to 20 nanometer in XY and Z up to 50 nanometer. So on the upper part you can see there is two protein which is called NUP153 and TPR. Both are part of the nuclear protein complex. So if you want to understand the arrangement of these two molecules along the nuclear membrane, so it is very difficult to understand with a normal microscopy technique, because if you try to after capturing the image, if you want to go into that level by zooming it, everything become pixelated. It is not possible. But if you capture a storm, you can see it is very easily visible that these two proteins are not localizing with each other 100%, but there is some region where they come in contact with each other, but there are regions where they are totally separate. Okay, so that kind of details is possible with the storm microscopy, which is not possible with normal microscopy technique. So in summary, we have already discussed about bright field contrast technique like phase contrast, DIC, and dark field. Then we discussed about white field fluorescence microscopy, how it is advantageous and what are the disadvantage, and how the confocal microscopy can solve this problem. Then we also discussed about super resolution microscopy, through which you can break the diffraction barrier and you can go beyond to capture super resolved image. So that is all from my side. And so here one image is like lower part is a confocal, or I think white field. Upper part is a sim image, that mitochondria, and you can see the nucleus. So now if you have any query we can discuss, or yeah. I hope no one is sleeping. I don't know, maybe too much information in very short time. But
Yes.
probably if you keep in mind some information, there is a very good site, it's called Nikon Microscopy U. You can go there and you can search in Google and you can see the details there.
So one question I want to ask to you: what is the use of AI in confocal?
Okay, so actually Nikon introduced this functionality recently. Now actually there are multiple AI functions available. So one example I can give you. So I showed you the white field fluorescence where a lot of out of focus light is coming. So with the AI function, you can train the software in such a way, or it is already trained, it will automatically understand what is the out of focus information and what is in focus information. From the white field image, it can discard the out of focus information, and it can keep only the focus information, and it can improve the image quality. So that is how AI can help you. And there are many other AI function like segment.ai, with which you can segment very difficult structure, which is normally very difficult to segment with the conventional tools available. And there is other AI tools also available that can help you to process the image for downstream segmentation and for to downstream analysis. So that is how AI can help. uh, any question now? If you have some time, I can quickly show you the advantage of AI. Just 1 minute, maybe. Let me open that. Okay, let me share my screen. Can you see my screen?
Yes, yes.
So this is called convert.ai. So let's say I have a bright field DIC image, but I don't have any nuclear stain. So I can train the AI to which can predict the position of the nucleus on a unstained sample where there is no staining available for the nucleus. So this is called convert.ai. You can see it is perfectly predicting the nuclear position. This convert.ai, enhance.ai, we can improve the, or we can predict from a very low exposure image a high exposure image, through which we can do a segmentation properly. And this one example where we have segment.ai. So let's say I want to measure the distance or some other parameters for this projection kind of structure, not actual the cell body. With the normal conventional tool, it is very difficult, because when you try to do the thresholding, it will always pick up this part along with the cell body. But with the segment.ai, what you can do, you can actually train the AI function to specifically pick up this segment, pick up this kind of projection kind of structure. So that is the advantage of segment.ai, which will help you for downstream analysis. And also we have denoise AI. So left side probably, let's say if your sample or is light is not very, or your sample is low signal sample. So if there is too much of noise, that you can take care with the denoise AI. So it will remove the noise part of the sample, you can see. And also this is another example of denoise AI, which can improve the signal, or the signal to noise ratio of the image. And this is clarify.ai, particularly made for wide field fluorescence microscopy. On the left side, I think these are Drosophila embryo that is searching for food. You can see this is a thick sample and too much out of focus light, and it is difficult to understand the structure, internal structure. So if you now apply clarify.ai, then probably you can see it is so left side is the normal image white field, right side is a clarify AI generated image. So it can show you the internal details. So that is how actually the AI can help you for different kind of segmentation analysis. Sorry,
Are all these AI can be used as a plugin in analysis software?
can you repeat please?
I'm asking whether all of these AI can be used as a plugin in Nikon software.
Yes, yes. I don't know what is the version of the software. Generally, the denoise AI come as a default, but other the AI function you can always add as a module anytime.
Okay, thank you.
Thank you.
Okay, if there is
So
no
in
more,
okay,
can we finish today's session? So tomorrow it will be physical. Uh, we will let you know as soon as the speaker is here. So it will be one of before lunch.
okay, okay. So I am leaving now. Thank you.
Thank you. Roman, thanks a lot.
Thank you very much.